First off, thank you, Bio, for this great opportunity, and also JLabs. Very happy to introduce our company today, Ticarus. We utilize the unique platforms for the next generations of cell therapy, especially CAR-Ts. So, starting with the mission statement, our ultimate mission is to develop a solid tumor targeting CAR-T. As you're familiar with, the solid tumor accounts for about 90% of all tumors globally. However, all the approved CAR-Ts and most of the clinical trials are focused on the hematological malignancies or blood tumor. It could be caused by the insufficient efficacy in solid tumors, cell trafficking, or tumor microenvironment that the characteristic of the solid tumor has. So the development of the solid tumor targeting CAR-T will not only open up a tremendous market, but also it will fill up the unmet medical needs today with the CAR-T therapy approach. So in order to target this tricky solid tumor with the CAR-T therapeutics, we have came up with three proprietary technology, clip, converter, and switchable. Just a schematic bird's eye view is that the clip is the better CAR backbone, which means that we have utilized with our transmembrane domain backbone for the efficacy enhancement. Converter technology, we have switched or converts the negative inhibitory CTLA-4 signal into a CD28 stimulatory signal. Switchable CAR, which we have recently published in the Nature Communication, allows us to have a controllable toxicities and dosing regimen. So just a bird's eye view of our click CAR backbone, which we'll be focusing for today. So three different single-chain antibody, which is the binding domain. Our leading asset in the clinical trial phase 1 is currently showing 100% complete regression for both in the low and the middle dosage, being conducted in the 6 big hospitals in Korea. TCO91, which is the CAR-T targeting solid cancer, is showing the robust in vivo efficacy and also the controllable toxicity that we are enabling our IND study and expected to file an IND sometime next year. I have not disclosed this target for this presentation, but we are ongoing partnership with the partnering company, and also we have validated a strong in vivo anti-tumor activity as well. So three of the single-chain antibody were validated, and also different modalities such as NK. We are collaborating with this Australian company Carterix, and recently we have seen a promising in vitro differentiation and cytotoxicity, and we expect to co-file the patent by end of next month. So that's very encouraging for us. So let's get into the fun part. So this is the basic schematic construct design for the CAR-T, binding domain, hinge, transmembrane domain, and signaling domain. And the global effort has been going on with the modification or the addition of the signaling domain, such as CD20 by the Yescarta Gilead, or the 4-1BB by the Novartis' Kymriah. However, not many effort has been going on with the modification of the transmembrane domain. So that's where our company has found our niche, and our leading scientists have identified this very unique transmembrane domain that could potentiate the immune synapse when the CAR-T is carrying out its therapeutic activity. Quickly about the immune synapse, it's a binding region between the lymphocytes, such as T cell, and the cancer cell or the antigen-presenting cell. And just by switching the transmembrane domain into our clip technology, we have seen or demonstrated a tighter and a wider cytoskeleton rearrangement. In other words, the immune synapse for the clip CAR-T cell to carry out its therapeutic activity. First mode of action data we have seen is that when the clip was applied, we have seen a very well co-localization of the actin and tubulin, which are two very important components for the cytoskeleton rearrangement, whereas the non-clip has shown a poor co-localization. This time we have seen a direct relationship between the clip CAR-T cells and tumor cells with the comparison drug group of the Novartis' Kymriah backbone. We have seen that not only the co-localization of the actin and tubulin, but we can almost see the migration of the actin and tubulin towards the cancer cell, which means that it binds stronger and the engagement longevity was enhanced as well. Just a quick progress update of our clinical result in phase 1 with the TCO11. We have seen in the low dosing regimen group both 1 and 3 months complete responses, as well as middle dosage. We have seen a 1 month response with the 3 complete responses. And so overall, we have 100% complete responses, and it looks like our third month responses as well will be similar to our previous one. So that's very good milestone that we're hitting right now in our company. So with this very interesting platform, we have chosen a very unique target called MUC1. The CAR-T, as I mentioned earlier, the CAR-T has many hurdles for the CAR-T therapeutic approach to solve. And one of them is the on-target off-tumor toxicity. On-target on-tumor toxicity could be somewhat alleviated by the antibodies like tocilizumab. But on-target off-tumor toxicity is still a big problem or the homework that we need to still address. And in order to address the on-target off-tumor toxicity, let me quickly mention the background of this MUC1. So MUC1 is a peptide that is overexpressed in various solid tumors, such as breast, ovarian, and pancreas. In the normal cell condition, however, so this blue line that I'm addressing right now is the backbone of this peptide, and it has a variable number tandem repeats, VNTR, which repeats the target itself from 25 to 125 times. So that means there's a rich targets or rich domain partners existing in the backbone of this peptide. However, because in the normal condition of the cell, the condition of being a hyperglycolization, which means there's a glycolyzed condition, is widely expressed in the mesh-like structure in the epithelial region of this backbone of this peptide. However, in the tumor condition, the state of being the hypoglycolization, which means a decrease of the glycolization in this peptide, allows us the binding partners to bind easier and penetrate more efficiently to this target backbone. And this is why we have concluded that targeting the MUC1 backbone can overcome the on-target off-tumor toxicity. Quickly, I would like to mention that all of the in vivo models that we have and all the assays that we conduct are 5 times lower dosage, which means that in the conventional CAR-Ts, 5 times less is considered as the subtherapeutic dosing regimen group. But however, we always conduct our studies with the 5 times lower dosage, and as you can see in the breast cancer and stomach cancer, even with the low dosing regimen, we have seen a almost complete regression and a complete regression for stomach cancer. Also, in the pancreas cancer as well, we have seen a complete regression even with the 5 times lower dosage. And based on these promising results, we are now generating and manufacturing a viral vector for our IND filing. I'll go over this quickly. This is our partnering company's target, and they are targeting the ovarian therapeutic CAR-T, and also this single-chain antibody has seen the validation of the CLIP as well. Next is actually my favorite technology of my company called converter technology that I had mentioned in the earlier slides. So normally, as you're very familiar with, the CTLA-4 is the immune checkpoint, such as PD-1, PD-L1. And when it gets bind to the B7, it actually inhibits the therapeutic activity of the CAR-T cells to inhibit the activity. However, the ideal situation is to have a CD28 bind to the B7 that could eventually activate the CAR-T cell to carry out its therapeutic activity. However, in the tumor microenvironment, the CTLA-4 and the B7's binding actually gets much more stronger, about 50 to 100 folds more. So what we did here is that we have positioned, or we have kept the CTLA-4 in the extracellular binding domain to harness the binding affinity between the B7 and CTLA-4. However, we have positioned the CD28 in the intracellular signaling domain, which means that even if the B7 gets bind to CTLA-4, it actually converts that signal into an activation signal. So that's why we have called it a converter technology. With this platform, we have chosen a B7H3. I'm pretty sure that you guys are familiar with this target in order to solve the tumor antigen heterogeneity, being the characteristic that it is overexpressed in the solid tumor, but also simultaneously in the tumor stromal cells, such as the intratumoral vascular endothelial cells. So I'm just going to show the in vivo data here. As you can see in the bulky breast tumor. You can see the interleukin and the converter combined has shown the complete regression, whereas the interleukin itself releasing CAR T has not reached a very ideal results. Also, many questions has been asked regarding our converter technology and regarding the hyperactivation, and that is one thing that we need to consider as well. But we have seen a bit of a loss of the weight, but we have seen a transient recovery towards the end of the therapeutic approach. So we have concluded that it is a controllable toxicity. Just to recap, we have a phase 1 TCO11 CD19 targeting CAR T lymphoma indication. We have now recruited, I think, second patient for our high dosing regimen group. So we'll be finishing up our phase 1 by third quarter this year and going into the phase 2, and hopefully to get marketed within 2 years. TCO91 is actually the hot topic right now when I'm partnering with the various potential partners. And because it's a CAR T that actually targets the solid tumor and showing the promising results, albeit in vivo so far. We do have a novel autoimmune disease targeting bispecific antibody, which I have not brought for this presentation, but if you guys are interested, please let me know. I'll be happy to go walk through the asset with you. So some of the notable activities that we have, as I mentioned, the Carterix. Carterix, if I may mention a bit of a history of their company, they had a history of licensing in one of the Korean CAR T product with a decent amount of value. So we're expecting, hopefully, the same result from this collaboration. We have seen the initial in vitro data which were reproduced with a reproducibility of maybe 3 times with the large scale, and we have seen a constant promising result. So hopes are high in my company. City of Hope, as you know, is a major institution or hospitals in Korea. They actually had wanted us to provide CLIT technology for their glioblastoma targeting CAR T. Last year, we were nominated as part of the J&J initiation, JLabs Korea. And it has been tremendous help from the global to domestic support from JLabs Korea. So thanks JLabs, we love you. And the USM institution, I cannot disclose the full name at the moment. We're ongoing clinical partnership. It's called the Cancer Focus Fund, and they'll be funding our clinical trials conducted in the US. So hopefully that goes well. Recently, we have published a manuscript in the Nature Communication regarding our switchable, and also CLIP paper is coming very soon as well. Thank you very much for your time, and if you do have any question, I'll be on the back of the room. Thank you.